So today we're going to talk about prokaryotic cell division, why cell size is important, and review sexual and asexual reproduction. This is lessons 4.1 and 4.2 in chapter 4 of the textbook, or unit 4 of the biology course. So I had students research spontaneous generation and biogenesis. Those are two different theories. The theory of spontaneous generation was disproven. It was a theory that living organisms come from non-living things. So at one point in time in the 1600s, people believed that mice came from wheat or flies came from meat. And a lot of that had to do with their lack of understanding of where living things came from. So Reddy and Pasteur set up controlled experiments that proved the theory of biogenesis, that life comes from life. So these are two scientists you want to know. You want to read about their research and how they went about a controlled scientific experiment. If we take a look at two methods of reproduction, we first look at sexual reproduction, and it's very important that you understand the proper vocabulary for sexual reproduction. This will introduce us to why we're studying cell division. So all parents produce, males produce sperm, females produce eggs in sexual reproduction. Proper names, the male produces sperm and testes, females produce eggs and ovaries. When that sperm and that egg unite, that's called fertilization, and a zygote or a fertilized egg is formed. So this process is combining genetic information from two parents, and it results in an offspring that's genetically different. So that has some advantages and some disadvantages. So I have those listed here. One of the greatest advantages is the offspring are going to be genetically different than their parents. That's going to be an advantage when to endure change and with evolution. The disadvantages is it requires a lot of time. There's disease, it requires energy, and it's a slower population growth. So what you're going to notice about asexual reproduction is the advantages and disadvantages are now flipped. Asexual reproduction, typically our prokaryotes reproduce this way. So here's a picture of binary fission, a prokaryote. Its DNA is kind of just looped here, no nucleus. Remember, it's a prokaryote. It's the duplicated genetic information, and then virtually it splits into two genetically identical copies. So in this case, this is a form of asexual reproduction, and this is how we get new organisms here. So students are supposed to read section 4.2 about 3 different types of asexual reproduction, because it's not just binary fission, there's also fragmentation and budding. Comparing and contrasting, students are responsible for comparing and contrasting the advantages and disadvantages of sexual versus asexual reproduction. So you should feel confident and comfortable with these terms. And we talked about the words like genetic variability. Genetic variability means differences. The word facilitate, it means it helps it along. So it helps organisms adapt. The word courtship is a common word. Students don't know what that means. Courtship is kind of like dating. So a disadvantage of sexual reproduction is courtship takes a lot of time and is resource consuming. Whereas in asexual reproduction, it's a non-issue. And the other word that students ask about is retards. Retards means to slow down evolution. So because these are all alike, there's no genetic variability. The asexual reproduction will slow evolution. You won't see a lot of change. Some ways that prokaryotes can sexually reproduce is a process called conjugation. And then endospores is another way to protect DNA and survive for long periods of time. So sometimes people say, well, what keeps prokaryotic growth in check? What keeps us from being taken over by prokaryotes, since asexual reproduction can happen so quickly? Well, prokaryotes growth is kept in check by things like food, space, resources, and then just waste accumulating. So bacteria are not going to grow and if you have these conditions are limited. Okay, and then the last concept I want to just touch base on is that a cell is going to divide so that it's small. We did an activity about cell size that deals with surface area to volume ratio. We just need to make sure we reduce information overload and reduce cellular traffic so that a cell can be efficient.